Welcome to BrainMatters, the show about all things neuroscience. Whether you're a seasoned academic or just curious about the brain, our guests will be covering everything from cutting-edge research to practical tips for improving our mental health. I'm your host, Thomas von Ryan, and today I'm joined by James Gattuso, PhD candidate at the Florey Institute in Melbourne, here to discuss the science of psychedelics and the default mode network. James, welcome to the show.
Thanks for having me, Thomas. It's a pleasure to be here.
Could you start by telling us a bit about yourself and the path that your academic career has taken over the years to get where you are now?
Yes, as you mentioned, I'm a PhD candidate, so I'm currently in my second year of a PhD. So I've done my undergraduate in a Bachelor of Science with major in neuroscience, and then I did an honors year in neuroscience. And I've been fascinated by the mind and the brain for quite a while now. I have some interests, some personal interests in meditation and philosophy and, the nature of reality. And once I discovered that the brain is the most complex object in the universe, I it piqued my curiosity as someone that loves to learn and kind of likes to challenge themselves. So yeah, I majored in neuroscience, and then as I got a taste of research in my honors year, where I looked at ketamine administration in a mouse model of OCD, obsessive compulsive disorder, I thought, although it was a really challenging year and kind of required a lot of late nights, I did fall in love with research and being on the cutting edge and trying to discover things that have not been discovered before and how that can hopefully help reduce the suffering of humanity. So, and also my interest in the mind and the brain led me to be very fascinated by psychedelic substances, drugs that produce profound alterations in an individual's perception, um, and what therapeutic efficacy they could have.
Amazing. You touched briefly there on OCD models and potential psychedelic use there in the model, to
Yeah.
clarify.
Yeah.
Could you describe how an OCD model in mice works
Yeah.
and what you were looking into there?
So there's a few ways that we can create a mouse model of a neuropsychiatric disorder. We can do behavioral interventions that could induce a sort of deficit. So, for instance, with anxiety or depression, we can chronically stress the mice to induce an anxious or depressive-like phenotype. And there's other interventions that we can do like that, or we can do a genetic mutation using CRISPR-Cas9 technology. And that's what has happened with the mouse model of OCD that we used. It's called the SAPAP3 knockout mouse model. So it's the most validated and researched mouse model of obsessive compulsive disorder to date. And SAPAP3 is a postsynaptic scaffolding protein in excitatory that's related to excitatory synapses. And when you delete this protein, these mice have aberrant excitatory neurotransmission, and they also display aberrant behavioral phenotypes. So, for instance, these mice will display excessive anxiety like behavior, which is also excessive anxiety or increased anxiety is seen in individuals with obsessive compulsive disorder. And these mice will groom themselves to the point of lesioning themselves. So, it's a harmful behavior that causes injury to themselves, but they cannot stop it, which is indicative of it being compulsive in nature. So it's important to understand that this mouse model doesn't, it mimics aspects of obsessive compulsive disorder. Mice aren't humans. And so the higher order cognitive components of obsessive compulsive disorder, like the obsessions, really can't be mimicked that well in rodents, but we can mimic some aspects like the compulsive motor phenotype.
And what did you find during that research?
Yeah, so we found that ketamine administration didn't really exert any anti-compulsive effects, which is ketamine's primary efficacy seems to be in with treatment-resistant depression. And ketamine is an interesting drug. It used to be at higher doses, it's an anesthetic, and it's still used in some cases for anesthesia. But at lower doses, it was seen to reduce suicidality in people with treatment-resistant depression. So we wanted to see how it would affect individuals with obsessive compulsive, a model of obsessive compulsive disorder. It hasn't been trialed clinically for obsessive compulsive disorder. not that I'm aware of. And yeah, we didn't find that it affected their behavior. Whereas when we looked at psilocybin in the same mouse model, and psilocybin is the hallucinogenic compound in magic mushrooms, we found that psilocybin did lead to significant and lasting reductions in that excessive grooming behavior, which is very, very interesting. And the fascinating thing about the reason why preclinical investigations have some advantages over clinical investigations, looking in human populations, is that we can really see how much of this is a true biological effect. So there's no expectancy effects in the mice, there's no placebo effects. We use a control, but the problem with clinical psychedelic studies is that even when the individuals are blinded to the treatment, they can still know if they're hallucinating or having an experience, an alteration in consciousness or not. So it is harder to blind. And so presumably some of the benefits could be coming from that. Additionally, there's also psychotherapy that accompanies the psilocybin experience in some of the clinical trials. So there's a hypothesis that psilocybin or psychedelics will create a state in which the mind will create this state in which the brain is more open to the benefits of psychotherapy. But however, in mice, that's clearly not the case. So we're really looking at a biological effect, and we can tightly control parameters, because ethics allows us to do so, and we can control more variables in a preclinical environment that can't be controlled in a clinical environment most of the time. So to see that effect was quite staggering and very exciting. And we're following up with what molecular changes occurred in the brain as a result of that psilocybin injection, and how that correlates, or how that may mediate the anti-compulsive behavioral effect.
Okay. So this was previous research on ketamine and OCD mice models. What are you working on at the moment as the big part of your PhD?
Yeah, so as I mentioned, that psilocybin and the OCD model wasn't a chapter of my PhD, or an aspect of my PhD, and I also then looked at psilocybin in 5-HTT knockout mice. So 5-HTT is a serotonin transporter, and so these mice lack the serotonin transporter, and they've been around for over 20 years as a model of anxiety and depressive like behavior, but they're also a model of serotonin dysregulation as well. So we wanted to see how psilocybin affected this mouse model. It was very interesting. We found that psilocybin will produce a head twitch behavior in mice. So this is a proxy of the hallucinogenic experience in humans, and we know which receptor mediates this, which is the 5-HT2A receptor, because in humans, when we block the 5-HT2A receptor prior to psilocybin, it abolishes the hallucinogenic experience. When we do the same thing in mice, it blocks the head twitch behavior in mice. So we can see it's very translatable, and we know exactly what receptor kind of mediates it. And we found that psilocybin didn't induce any head twitches in the 5-HTT knockout mice, as opposed to the wild type mice, which it did do the standard head twitch response. And we also found it didn't have really any effects on any other behaviors in the 5-HTT knockout mice, but it did exert some effects in the wild type mice. So it seems that the serotonin dysregulation of this mouse model meant that psilocybin couldn't quite bind to the receptors in a way that would exert the expected behavioral effects. So that's been interesting. It's a little bit technical as to why that may be the case, which we're further exploring. But yeah, it kind of suggests a few things: that the serotonin transporter may be involved in psilocybin's efficacy, but also that this mouse model, but due to dysregulation of certain receptors in this mouse model, psilocybin may not be able to exert its effects, which kind of is revealing a little bit more about the neurobiology of psilocybin.
Now, these are mice models, obviously limited.
Mhm.
Can you foresee how this could be researched in people in the future to understand it better?
Yeah, so there's a plethora of clinical studies going on at the moment investigating psilocybin in a variety of different neuropsychiatric conditions. You've got anxiety and depression, obsessive compulsive disorder, anorexia nervosa, post-traumatic stress disorder. So there's a lot of studies going on at the moment for the potential efficacy of psilocybin, and then the classical psychedelics more broadly. So I think there's a variety of ways of looking at it. Ensuring that there's an appropriate placebo and blinding is very, very important in psychedelic studies, and whether there's one the dosage and the number of dosages are an important factor and variable. The age of the population is also very interesting. Usually, it's been done in adults for ethical reasons, and various reviews have looked at the mean age in which they do look at studies. I mean, look at patients, but actually, a more important factor is their prior psychedelic use. Whether they've had experience with a psychedelic or psychotropic medication before. These are all important variables that could influence outcomes. However, there's a plethora of studies going on at the moment. Some are looking at using fMRI to understand the neurobiology of what's going on in these various patient groups. Some are using EEGs, which measure the electrical activity on the scalp, and some don't necessarily are more looking at clinical endpoints. Some are looking at safety. So yeah, there's a lot going on in this space.
Now, you touched on the fMRI research
Mhm.
there. It seems you can't really talk about psychedelics now without discussing the default mode
Mhm.
network. What is this network and why has it become so ingrained in psychedelic research?
So the default mode network it's a cluster of brain regions that are quite midline if you do a cross section of the brain, but it's a cluster of brain regions that are involved in this sense of self. When it's called the default mode network because when an individual is not doing anything, there seems to be enhanced activity in these brain regions when looked at under an fMRI scanner. And these brain regions to be correlated with self-referencing and mind wandering and notions. So when an individual is kind of just minds wandering, and often they'll think about things in relation to themselves, thinking about the future and thinking, what am I going to eat for dinner? What am I going to do after this? These brain regions seem to mediate that sort of thinking. And there seems to be alterations in the default mode network in a number of neuropsychiatric conditions, but specifically depression. In depression, there seems to be excessive activity in this default mode network. And that makes sense because an individual with depression can often think a lot about themselves in a very negative context. I'm unworthy. They can think of, I'm not good enough. And it can be a lot of, thinking about themselves in a negative valence. And researchers have found that potentially increased activity in the default mode network could mediate that. So psychedelics have consistently been able to reduce activity within this network, but interestingly also increase activity between brain networks. So the brain becomes less modular and more globally integrated. So when I say less modular, the brain has different brain regions and different brain networks. And so you could think of the brain operate, there's the visual cortex that's involved in vision. There's the prefrontal cortex that's involved in higher order cognition. There's the somatosensory cortex, which is involved in touch and feeling. And so all these different brain regions are involved in various unique functions. But under the influence of psychedelics, these distinctions, the clear demarcations of these functions start to disappear, and the brain functions more as a holistic, integrated whole rather than this separate compartmentalized organ. And so what happens then is that this reduced activity in the default mode network and this increased activity between networks seems to give rise to this feeling of ego dissolution, which can happen, which reliably is invoked under psychedelics. And this ego dissolution can give rise to this feeling of oneness, this unity with all things, because once the ego starts to starts to dissolve, or this sense of self starts to dissolve, there is this feeling of oneness, feeling of connectedness. And that has been also shown to predict some of the therapeutic outcomes associated with psychedelics. And psilocybin, lysergic acid, LSD, dimethyltryptamine, DMT, these all act on the serotonergic system. That's why they're called classical psychedelics. And all of these psychedelics have been shown to have very similar actions on the default mode network, reducing that activity. And as psychedelics have shown to reduce depressive symptoms in patients with depression, the research suggests that this could be mediated by reductions in the default mode network, among other factors.
Thank you. Now, you said about classical psychedelics and how they are serotonergic or based on serotonin action, but there's also been lots of discussion around other neurotransmitters. You yourself have previously been involved with research looking into the chemical messenger glutamate. What prompted looking into it and what did you find?
So, as I mentioned, the classical psychedelics act on the serotonergic system, and ketamine is a non classical psychedelic. So, it's actually a dissociative, and it acts on the glutamatergic system, which is one of the things that separate it from the classical psychedelics. And it's specifically an NMDA antagonist. So it's interesting that although both ketamine and psilocybin may lead to reductions in depressive behavior, they do fundamentally act on different neurotransmitters. However, the classical psychedelics, such as psilocybin, for instance, they do increase serotonin, but then, they bind to serotonin receptors, but the end product of that is still an increase in excitatory neurotransmission. So they do still increase glutamate release in the cortex. And there's a very interesting study by Mason et al. In 2020 that found that was the first, because it had only been hypothesized that that was the case in humans, and also there was preclinical evidence using rodents, but they were the first to find that psilocybin does indeed increase glutamate. But they found that it increased, they looked at two brain regions. They looked at the medial prefrontal cortex, which is also one of these hubs that is very important in the default mode network and related to this higher order cognition and thinking about the future, and also related to this sense of self. And implicated in depression. So they looked at psilocybin, so how psilocybin impacted glutamate in this brain region. And they also looked at how psilocybin impacted glutamate release in the hippocampus, which is a key brain structure involved in memory and depression and neuroplasticity. And so they found that psilocybin increased glutamate in the medial prefrontal cortex, but this was also associated with actually the increased anxiety that can occur during the psychedelic experience. So during the ego dissolution, it's not always a positive experience. There can be anxiety associated with that experience. So they clustered individuals into two groups: people that had an anxious ego dissolution and people that didn't, more of a euphoric ego dissolution. And they found that this increased glutamate in the medial prefrontal cortex was associated with that increased anxiety response with the ego dissolution, whereas decreased glutamate in the hippocampus was associated with the more positive ego dissolution. And that's quite consistent with what's known about glutamate's function. So if you look at anxiety, for instance, anxiety is generally characterized by excessive neural activity in the brain and a hyper glutamatergic state. And that's why if you look at the anxiolytic drugs, they generally reduce neural activity in the brain, and they generally increase GABAergic neurotransmission, which is the main inhibitory neurotransmitter in the brain. So it kind of balances that excessive activity back down to normal levels. And so, yeah, that's been the only study, to my understanding, in humans that's looked at psilocybin and glutamate release. So so the psilocybin, I mean, because serotonin is a broad neuromodulator, it interacts with a lot of other neurotransmitters. So it interacts with dopamine, for instance, and it interacts with glutamate. And there's also serotonin receptors on GABAergic interneurons. So psilocybin can also increase, yes, excitatory drive, but it can also increase inhibitory activity in the as well. And this broad effect on a range of different neurotransmitter and cell types produces a sort of symphony of activity in the brain that drastically kind of changes the brain state, and as a result, drastically changes an individual's perception or of reality.
Perfect. Before we wrap up, I would like to ask you some light-hearted
Yeah,
questions to give people more of a glimpse at the man behind the scientist, so to say. Does that sound good?
yeah, sounds great.
Have you been watching the Olympics?
I actually been so busy that I've only just been kind of briefly watching the news about it, and Australia are doing pretty damn well. So, but I
They're doing very well.
haven't been following it too deeply yet.
What's your favorite event?
You know, I enjoy watching the ping pong, to be honest. Yeah, yeah, because I like playing ping pong myself, and I think when you play a sport, you've spent time engaging with that activity. You can just appreciate how good the elite people are, right? Like, I haven't done horse
Definitely.
riding, so I really can't appreciate the equestrian to the same degree I can with something like ping pong. So ping pong's pretty amazing. I think also the 100-meter sprint, you can't look beyond that. So I know you said one, but I'm gonna pick two.
Universally enjoyed,
Yeah,
definitely.
yeah.
Is ping pong one that you could imagine yourself doing if you were to go to the Olympics?
Yeah, I would say I'm not too bad at ping pong, so that you know, I think I could. And it's actually engages a lot more cardio than what people think if you engage in serious ping pong.
Are there office ping pong competitions at the Florey?
There are, but I don't want to ruin someone's day, so I don't participate in.
On that topic, are there any hobbies that you like to spend your time on the weekends?
Yeah, I've got quite a few hobbies, so I do my best to manage everything to have work life balance. But yeah, I love to exercise. I love to go to the gym and lift weights. And if I can't lift weights because my shoulder's hurting or my back's hurting, then I'll reluctantly do cardio. And I love to read. I love to read of various different domains. I like to read. Some fiction. My favorite book is The Karamazov Brothers by Dostoevsky. And, I read some nonfiction as well. I like to read, general science things in other domains. I like to read some philosophy, some spiritual books. Yeah, that covers me.
Amazing. What book do you have on rotation at the moment?
Yeah, oh, that's a great question. I just finished. I can't remember the exact title, but it was on alcohol by Professor David Nutt, who was a co-author on my paper in the Default Mode Network, and he's a leading neuropsychopharmacologist in the UK. And it's just a new science of alcohol and the health effects, which is very interesting. And now I've started reading A Brave New World by Aldous Huxley.
Finally, what is the best advice you've been given by someone that you respect?
Mm. With effort comes reward. Yeah, and that was by my dad. Yeah.
Lovely.
Yeah.
Okay, James, I think we've covered quite a lot, and you've given people a very good introduction to your work in the field of psychedelics. While you have the platform, is there anything that you or your lab is doing that you would like to plug or promote?
In terms of our lab, if you search Anthony Hannon on Google Scholar or Thibaut Renoir, Thibault Renoir, Renoir, on Google Scholar, you can have a look, or on Google, you can have a look at what our lab is up to. We're doing a lot more than just psychedelics. So our lab is looking at all sorts of different things, epigenetics and transgenerational effects of infections and COVID, which is very, very interesting, and looking at the gut microbiome. So there's a lot going on more broadly in our lab. For myself, you can just search James Gattuso on Instagram or Facebook if you want to direct message me.
And if people wanted to reach out to you either for collaboration or discussion, you mentioned your socials. Is there anywhere else where they could do so?
Yeah, my email is also J Gattuso, Gattuso, at student.unimelb.edu.au. So if you want to send me an email.
Amazing. James, thank you so much for your time
Thanks,
today.
Thomas. Thanks for having me. It's been wonderful.
If you enjoyed this discussion and want to learn more about the brain, why not check out our website at brainmatters.nl, where you can find easy to follow articles in both English and Dutch to satisfy your curiosity. Until next time.